Alright everyone, let's work through an example of a Hardy-Weinberg equilibrium problem. When you see these on the exam or you see these in practice, remember that these will be in the form of word problems. So take some time, make sure you read the word problems, and make sure you figure out the context that you're going to be working through. So the important terms were listed above this video, so make sure that you account for all those terms when you're reading through the word problem. Okay, let's look at this example here. So we have these two different varieties of blue-footed booby, which is a type of bird, and the specific phenotype we're looking at are webbing in the feet and not webbing in the feet. So first thing you have to do is determine what is the dominant phenotype, what's the recessive phenotype. In this slide here, I basically told you the answer to that. So if we look at the phenotypes, you can have no webbing or webbing. So which one is dominant and which one's recessive? You should be able to figure that out by looking at the genotypes. So given that we have the dominant phenotype being no webbing, you can either have a homozygous dominant individual in terms of a genotype, a heterozygous individual that both would have the phenotype of no webbing. And the only way an individual could have webbing on the feet is if they were homozygous recessive. So these terms from our last unit on genetics are going to come back to haunt you again. So make sure that you know what these terms mean. Okay. So here are three different types of genotypes. And in this case, let's say we're working with 500 different individuals. So we have a population of 500 animals. 320 of them have this phenotype and this genotype, big W, which is homozygous dominant. 160 individuals have the heterozygous genotype and no webbing, and 20 individuals have the homozygous recessive genotype and webbing. So all of Hardy-Weinberg works through frequencies or proportions. So we're working with decimals or numbers less than 1. So we have to first figure out from the information given in this word problem what are the allele frequencies. Remember, allele is P or Q, the dominant allele or the recessive allele. Keep in mind that each individual has 2 letters in their genotype. So we have 500 individuals. We have to multiply that by 2 to determine the number of alleles in the population, because these are diploid individuals. So 500 animals, but 1,000 alleles. This also makes our math pretty easy at this point. So 320 individuals have this genotype of big W, but there are 640 big W alleles in these individuals. Let's skip over to the webbed example. There are 20 individuals with this genotype of homozygous recessive, meaning there are 40 little w alleles. So what about this group in the middle? The group in the middle are heterozygous, meaning 1 big W, 1 little w. So you take 160 having big W, and you have 160 little w's, and you add them together here. If we're looking for allele frequencies, meaning the number of P's and the number of Q's, P here is the dominant allele, which is the same thing as a big W. So how many big W's exist in this population? Remember, the denominator here of 1,000 is the total number of alleles in our population. We have 800 big W alleles, 640 from these individuals, and 160 from the heterozygotes. So 640 plus 160 equals 800. 800 over 1,000 is 0.8. So our allele frequency of the dominant allele, or P, is 0.8. By math, you can subtract 0.8 from 1 and get the recessive frequency, which is 0.2, Or you can do it the long way and add 160 little w's from the heterozygote to the 40 little w's from the homozygous recessive individuals. 160 plus 40 is 200 over 1,000, or 0.2. Now we have our allele frequencies. We have a P, or dominant allele frequency, of 0.8, and a recessive allele frequency of 0.2. Write those numbers down before we move into the next phase. This is phase 1. Phase 2 is determining genotype frequencies. Remember, a genotype is 2 letters. And this is where our terms homozygous dominant, homozygous recessive, and heterozygous come in. If I ask you to determine the genotype frequency, you take your allele frequencies from the last slide, 0.8 or 0.2, and simply plug them into this equation. So 0.8 squared gives you your homozygous dominant. 0.2 squared gives you your homozygous recessive genotype. And then 2 times 0.8 times 0.02, or times 0.2, sorry, 0.2 plus 0.8 times 2 gives you this middle number, which is all worked out here for you. Here are our allele frequencies, 0.8 and 0.2. 0.8 squared is 0.64, so our genotype frequency is 0.64. Our recessive, homozygous recessive frequency is 0.04, because 0.2 squared is 0.04. And then to determine the heterozygotes in the middle, it's 0.8 times 0.2, which is 0.16, times 2, which is 0.32. So that's really how you solve Hardy-Weinberg equilibrium equations. You determine allele frequencies, or the frequency of P and Q, the dominant versus recessive, and the genotype frequencies using the longer equation. And so you must write down all of the context given to you in any of the word problems, and then simply plug and play from that point on.